So just another small adjustment to the schedule. Tomorrow morning we're going to begin with Renee. We're moving Renee from the end to the start of the meeting tomorrow. This doesn't affect the timings. And then the two partner talks will be from 12:30 to 1 in the place of where Renee is now on your programme. So with that, let's continue with the AI computation session and have Jane Osbourn from who's the CSO at Alchemab, mining the immune system for patient-derived protective antibodies. Jane.
Thanks. Okay, well, first off, again, thanks to Antonio and the Fair Journey team for putting on such a wonderful conference. It's a beautiful place, but it's also a really great community. And I think this community is going from strength to strength. And at the end of the day, that's what drug discovery is about, is building connections. So thank you for that. And I feel particularly privileged that I'm actually speaking for the second year in a row. So hopefully, I'm very happy to do 3 in a row, if at all possible, because this is just such a great meeting. So Dennis was reflecting on the boat yesterday about what made him feel old, and that his children were old. And I was reflecting on the same question and realised that, well, I've actually done 30 years of antibody drug discovery, which did feel like a rather long time. But it's been a really interesting time. You know, I started before many of the drugs were approved. We've now got 120 monoclonals on the market, and I've had a really fulfilling career. But most of that career was working in target-driven drug discovery, using all sorts of different technologies, phage, ribosome, transgenics, cell sorting. That's been very productive, very successful. I work with some great people, and we've actually got a number of antibodies onto the market. But as I was sort of thinking through sort of latter few years, I became increasingly concerned that we were all working on the similar target space, and that there was a real need for some genuine innovation in drug discovery to give us some different insights into target biology. The other reflection that I had from my time at MedImmune AZ was pharma tend to focus on ill people and trying to understand how they can make them better and how they can interfere with disease pathways. And I actually did a little bit of work on digital monitoring of patients and started to think, well, there's a yin and yang to everything. Why aren't we looking at people who are well, who were sort of unexpectedly well, and is there anything that we can learn from that? So all this thinking grew into a point where I decided to leave AstraZeneca and start Alchemab, which is really focused around those themes. So antibodies from resilient individuals, mining them to see if we can find therapeutics. One thing that, so I haven't got a corporate slide on Alchemab. Just very quickly, we were founded in 2019, seed funded by SV, founded with nothing other than this idea, which was a bit scary. No IP, no assets, no samples. And we went from there to a Series A led by RA Capital in 2021. And we're now 60 people, mainly in Cambridge, UK, with a small neuroscience lab in Boston. So that's where we're at. So what I'm going to do is tell you a bit more about this concept of mining well people. It's all very well having a concept. You've got to turn it into reality. So how have we built the platform? And then what are we starting to see coming out of the platform? And it's nice to be here a year on and actually have some really solid examples of what we're finding. And then I'll summarize. So the concept, kind of already explained this, but we start with resilient individuals, cohorts, that's important, not just one, a group of individuals who have some interesting resilience to disease. We then deep sequence their heavy chain repertoires at as deep a level as we possibly can. we look for antibodies that are shared in those resilient individuals and not seen in disease progressors. And then we actually try and work out, well, what are those antibodies doing? What are they binding to? Do they have any functional properties? So this is a completely unbiased approach to drug discovery. We don't go in here with any preconceived ideas about the targets. We ask nature, what's your response to this disease condition? And how are you trying to protect these people? Not all these people are going to have any resilience because of the immune repertoire, but our sort of hypothesis is maybe some of them do. So resilience takes many forms. It can be long-term cancer survivors, patients with a genetic predisposition to a neurodegenerative disease that don't develop it or develop it slowly. And we've also got access to some supercentenarians who are very long-lived, never had any chronic disease, mentally really, really cognitively great. So our hypothesis is some of these people have protective self-autoantibodies. Now, autoantibodies, as you know, tend to have a bit of a bad guy kind of reputation. They do cause autoimmune disease, lots of different pathologies associated with autoantibodies. But there are also, if you look in the literature, examples on the yin and the yang of this, that there are protective antibodies out there. Shirley gave an example earlier. This is a similar one. A cohort of breast cancer patients, some of whom have anti-HER2 antibodies that have been naturally produced, and they have a really significantly improved recurrence-free survival compared to the patients that don't. But there are many other examples in the literature. The thing is, they're hard to find because people tend to be maybe doing a little bit better if they've got them. So that's the concept. So that's all well and good, but it's quite a difficult thing to turn that into a platform to start mining for these antibodies. And the approach that we've taken is to take a very integrated approach of using bioinformatics and machine learning tools with a lot of wet lab demonstration and iterating this and learning between the teams. We have a very integrated team structure. So we get patient samples, we do repertoire analysis, sequencing in the lab, but we have computational tools to mine those repertoires. There are various ways we use phage and computational to give us our antibodies, and then the target deconvolution predictive screening takes us into, hopefully, a really nice portfolio of MAB projects. The first thing is getting the samples hasn't been a trivial task. As I said at the beginning, we didn't have any samples. It was my job to go and talk to clinicians, tell them the story, and see if they thought this was a good thing to do with their patient samples, which was quite an interesting and very enjoyable thing to do, actually, because most people said, yes, that's an interesting idea. It's fairly obvious. Why hasn't anybody done it? So I managed to get, with the team, a really nice cohort of different types of organisations in oncology, in neurodegeneration, providing us with samples. And that not only gave us the samples, it also gave us access to the clinicians who helped define the resiliency criteria. And the dream is, we then take our antibodies back to those same clinicians and say, here, please can we now partner with you in terms of the clinical approach. So you've got to sequence a lot of BCRs for this to work. We tend to try and sequence around 100,000 to 1 million per individual. So say we have a cohort of 12 long-term survivors and 12 progressors. That might give us a sequence pool of around 10 million sequences. And from there, we look at, okay, how are those sequences grouped in terms of relatedness? We use sequence relatedness as our sort of workhorse, but we're building up paratope and functional relatedness as well. And then we look, well, how many of those are convergent just in the resilient individuals, which takes it down to a few thousand. And then we do a kind of expert triage of looking at, of those, how many of those are class switched, somatically hypermutated, and we whittle that down to choose a few tens of antibodies that we're actually going to make and put in the wet lab. Convergence is rare. I mean, Dennis again mentioned yesterday the theoretical diversity is somewhere around 10 to the 14 to 10 to the 18. But it does occur, and actually the next speaker, Brian, has done some really nice work sequencing billions of healthy controls and getting a feel for that convergence. And we need to take that there is a little bit of natural convergence that we can subtract in our process. So we choose our antibodies, then we make them. Now, the compromise we've made is we've got to do deep VH sequencing. We're losing the natural paired reads. And the way we've got around that is we've built our own proprietary data set of over 1 million naturally occurring paired reads. And we use that to train our transformer, our machine learning model, to give us the best option in terms of a paired light chain. We also have access to phage, VL repertoires, and we do some single cell sequencing as well. So we can sort of iterate on the light chains and build our antibodies from that. The real heavy lift we've done this year, and I was looking at the talk I gave last year, was target deconvolution is incredibly challenging. And in the end, we decided to sort of take the bull by the horns and build our own in-house human antigen library, which we've done in a display format. We actually use yeast display for this, and we use antibody capture to then capture the potential antigens from the antigen library. So it's a human orpheome, 3,000 proteins. And this is working really well. This has been a step change. And we complement that with the classic IPM, mass spec, protein microarrays we use quite a lot. And also, again, integrating the computational approach of looking for homologies that could give us an indication of target. And then we put those antibodies through various phenotypic screens. So cell binding assays, different iPSC-derived neurons, astrocytes, etc., MLRs, and we do more computational analysis in terms of structural predictions and functional predictions. So we're starting to build a picture of what's the target, does it do anything disease relevant, and then how can we sort of build out and use that information. So we think this is quite a differentiated platform because we're using cohorts of well people. We are able to now do this target deconvolution. We're finding a wide range of novel targets. And this integration of machine learning at every step, I think many companies do this very well in terms of focusing on specific steps in the drug discovery process. But we're really trying to bootstrap ourselves in terms of that wet-based knowledge as well. So, the proof of any platform is, well, what does it deliver? We ended up founding Alchemab just at the sort of start of the pandemic, which was really challenging. We were recruiting people without meeting them. Everybody went through this. And we also then started to struggle to think, well, how are we going to get samples? So we thought, well, why not just test the platform on infectious disease on, using COVID as a workhorse, see if we can get the platform working. And it's actually quite a nice, relatively technically low bar because you're expecting an acute immune response. And you've got a pretty good idea that you're going to get some RBD binders out of this if it works. So we did just that. We managed to access samples from around 30 patients who had recovered relatively quickly from COVID. We did have a few in there that actually went into more severe disease as a sort of progressor comparator. And we found a lot of convergence across this group of people. We published this in 2019. We also saw some interesting clonal expansions in the COVID repertoire kind of maps compared to controls. And we were able to demonstrate that we had indeed selected convergent neutralizing antibodies. Not all of them are neutralizing, but we have a nice panel of broad spectrum neutralizers. This was pre-Omicron, so these didn't get taken forward. This is compared to what was then the benchmark. I think these are the Regeneron antibodies. So this was really encouraging that we were seeing this nice convergence binding to RBD. About 50% of our antibodies bound to RBD, and then about, I would say, about 10% of them neutralized. So good start. What about oncology? So oncology, you've got a much more chronic response to the disease. So you're potentially thinking of, are we seeing convergence in samples that potentially, have been building for a few months to a couple of years in terms of the immune response. So this is a sort of, I would say, tier 2 of technical challenge. And we wanted to go after really hard to treat diseases, so we chose pancreatic cancer as our sort of first pilot here. We managed to set up a really nice collaboration with Oxford University, who had some long-term survivors, so 7 years, which is really, really rare for pancreatic cancer. Probably about 2% of patients live that long. And then we had a matched group of progressors, healthy controls. We ran our convergence funnel. We chose around 30 or 40 antibodies to make. And this just gives you a feel for the amazingly surprising level of convergence that we were finding in these long-term survivors. So this is the panel of our sort of top 10 most convergent antibodies. And you see these 3, 8 out of 12 long-term survivors having exactly the same antibody. And it's not a short CDR, it's 19 amino acids in the same germline with actual reasonable representation in the repertoire. Also, all of these, 80% of these were IgG, so they've been class switched. There was one IgA, and an interesting IgM with a lot of somatic hypermutation as well. So we've started deconvoluting these targets. We find, reassuringly, sort of clinically validated targets, PD-L1, CTLA-4, etc. But we're also now uncovering less well-validated targets. So there's a bit of a theme around the interface between innate immune targets, which is interesting given Shirley's talk. We found anti-complement factor H antibodies, which have a very interesting binding profile. They only bind complement factor H in the context of the tumour. So they take away it's kind of like an innate checkpoint. We found some interesting tumour cell targets which correlate with survival, and some interesting sort of known targets in novel settings for cytokines and chemokines. So this is all really encouraging. We're actually partnering a lot of the target validation for oncology because it's quite complex for us to set up assays for everything that we're finding. So if anybody's interested in sort of finding out more about that, please ask. But the reason we're doing that is because we decided we should actually sort of really go for the holy grail of sort of tier 3 complexity and see if we can find any convergence in neurodegenerative resilient individuals. So, and the main reason for doing this is actually, from my mind, this is the biggest vacuum of new biology and new targets that we need to do something about targets in neurodegeneration. So one example that came out of this is our preclinical candidate ATLX-1088. This is an antibody to CD33, which is a microglial receptor. So microglia are the inflammatory response in the brain. CD33 is an inhibitory receptor. It has a sialic acid binding site, and when sialic acid binds, this triggers the microglia to reduce their activation and lower their level of phagocytosis. So we found a convergent antibody in 2 individuals who had significant risk factors for Alzheimer's disease and hadn't developed the disease. This was in collaboration with the European Prevention of Alzheimer's cohort, and the clinician defined these resilient criteria for us. So we found this initially in Alzheimer's. We went back and screened our database to see if we could find any more of these antibodies, and we found consistently in the Alzheimer's and in the supercentenarians that we had antibodies that were binding consistently in this ligand binding site in resilient individuals. CD33 has a GWAS association with Alzheimer's disease. So there's mutations in the long form that predispose you to Alzheimer's, and there's a truncated mutation which sort of in some ways mimics the fact that we're blocking this ligand site, that actually gives you less risk for Alzheimer's. So this is a really nice, compelling biology story. And the antibody that we found actually has some really different biology from all the CD33s gathered by classic target selection. in that, well, first off, it does a great job of stimulating phagocytosis of the microglia. So this is our antibody. One of these down here is a competitor, Elector anti-CD33 antibody, so a really significant increase there. But also, our CD33 antibody doesn't internalize. So CD33 does internalize really rapidly. This is what happens from starting here. It's just too quick. But the competitor antibody immediately internalizes. Our antibody sits on the surface of the cell, just like causes no internalization. So this is great from a PK perspective, that we could potentially use this to treat peripherally. The microglia have the potential to migrate to the brain and phagocytose plaque load. So we're pretty excited about taking this forward. And then the second neurodegenerative target that we've found, I can't actually name the target, unfortunately, so this is the reason to come back year 3, by the way, is ATLX1282, and also some other emerging themes. So ATLX1282, we found convergent in 3 individuals that were mutated with progranulin or C9orf72 to give them a predisposition to developing frontotemporal dementia. It's a completely penetrant mutation, so by rights these people should have had FTD and didn't. In 3 people we found this antibody, again with class switch, nice somatically mutated, long CDR. It binds, what I can tell you is it binds a neuronal receptor, and it seems to protect the neurons from cell death in the presence of amyloid. And it also looks like, it's quite hard to see in this light, but it has the partial rescue of dendrite growth. So it looks like it's actually causing, resulting in some regrowth of neurons. So I'm really excited about this one because I think this is potentially disease modifying. And as I said, there's a real need for more approaches in dementia. The other themes that we've found from the targets that we've deconvoluted so far from the neuronal cohorts is neuronal health is definitely a theme. We found this already. There's a couple of other neuronal receptors that we're working up and looking at their functionality, and neuroinflammation. So I've talked about CD33. We found other targets related to neuroinflammation and some quite interesting sort of slightly off the wall enzyme inhibitors and transporters, which we're sort of trying to sort of position. It's hard to prove a negative, but it's just worth flagging that we have not found any anti-amyloid antibodies or any anti-tau antibodies in these resilient individuals. It may be that we're not screening appropriately for them, but it's just sort of, I think that's just, an observation worth sharing with you, because I do think, whatever the status of amyloid, and I'm really pleased that the amyloid antibodies are being approved, we're going to need combination therapies in dementia like we have in oncology. So we need a broader portfolio. So that's a flavour of some of the targets that we've found. We've also, on this journey, really developed some very nice machine learning tools which give us real insights into the data that we have. So we're now at a point where we've got certainly the largest resilience-associated database of BCR sequences, I would say, coupled with patient metadata, coupled with information about functionality of the antibodies. So this is we call it our data cube. This is our resource. And it's sort of fairly logical to think, well, what can you do with that resource? We can use it for sort of further target validation. So the CD33 antibody that I just described, we found that in some resilient AD individuals. You can then go back and look into the database and say, is this antibody seen in any other neurodegenerative conditions? So this is an example where an antibody's been found in FTD and Alzheimer's, largely associated with resilience or healthy controls. So you can sort of go back and mine and start getting maps of where these antibodies might be clinically relevant. We can also go back and just do a sort of classic mine of related sequences. This is really useful because you can then choose the best antibodies in terms of their developability profiles. And so you're sort of like gathering all the variants of a sort of parent sequence. And again, that's reassuring that you find a number of variants in the individuals. So I think sort of some kind of active selection's been going on where you can get these sort of phylogenetic trees of these antibodies. And then we've also developed a machine learning platform. And I should actually give huge credit to Jake Galson, who is in the audience somewhere, who's been leading on all the informatics and the machine learning. So this is called Antibody. It's an antibody-specific transformer model for representational learning of all the data. And it basically has a two-step approach. So step one is pre-training the model on a very large set of unlabeled data. And then step two, we fine-tune the learnings using labeled data, and we can apply this to various different settings. So this is interesting. So this is actually a representation of what Antibody gleaned in terms of information from that first step one using unlabeled data. Even without telling it anything, it can tell you which are the naive B cells, which are the memory B cells. It can recognize germlines. There's an interesting little cluster here, which maybe tells us something about germline structures and a number of mutations. So this is really exciting, and we are applying this already in the core platform. So we use this for the VL pairing, functional clustering on the convergence, and other kind of parameters. So we're interested in tissue localization, predictions, and things like that as well. And moving forward, I think there's many ways we can use this to support the development programs. It also is quite interesting that we can take the repertoire as a whole and see if there are any particular insights around patterns of antibodies in different disease settings that might actually help in terms of health insights. So that's so this is all underpinned by the huge data resource that we now have and this transformer model. So I'm doing all right for time. I've only got 5 minutes on the clock, so I will just sort of do a little bit of reflection and summary. So I'd say like that ultimately you have to demonstrate that your platform has value. I've spent a lot of time working in platform companies, and you have to get those first proof of concepts of what you're finding. And I'm really confident now that we're there. We're starting to build a really nice preclinical pipeline focused in neurodegeneration because we think we can do something different there. And I really want to make sure we're spending our time doing making the best impact we possibly can. But we have got oncology targets and a preclinical program coming through. And there's no reason why you can't apply this platform to any other disease where you want to define resilience. So we're also sort of thinking, can we do discovery collaborations in areas that aren't our core? So we have a nice evolving pipeline, potentially INDs next year for any of these, depending on in vivo data. They're all in vivo proof of concept models at the moment. Why does it matter? I do think we need to have more understanding of immunological response in health as well as in disease. And we've got some really interesting observations from the supercentenarians in terms of limited diversity of their repertoires, which I think is important. So overall understanding more immunological health. We have an absolute urgent need for new targets. We absolutely have to do something different. And I'm passionate about trying to solve that. So I think that is going to make a big impact, hopefully. And I think target agnostic approaches in general just give different hypotheses. I mean, it's not, we don't have to go and use the antibodies as the therapeutics. Maybe these give insights for small molecule approaches or gene therapy. But just having a different lens on how people evolve with their disease, I think is important. This might seem a bit grandiose, but I've worked with lots and lots of different technologies over my 30 years as a slave to drug discovery. And what I'd like to sort of try and do is complement all these wonderful technologies that have been massively successful with different ways to provide them with targets. So, you know, maybe this convergence approach using samples and you know, computational screening, it potentially gives you human antibodies at the end of it as well, but it also feeds back into, well, can we do more targeted campaigns on some of these targets using more target-driven approaches. And I think that's something that we could aspire to. So it's been a great journey. I mean, starting from nothing, we now have a relatively robust pipeline, and we're having a lot of fun. We have a wonderful team of people. I haven't put the sort of the leadership team up, but great group of people many of whom have been doing classical drug discovery, and we're up for doing something different. And I really appreciate the people that took a risk to join the venture. We call ourselves the Alchemabians. This is the team in front of our little lab in Whittlesford near Cambridge. So I want to thank all the team. I want to thank our many, many collaborators who've just been so enthusiasm is so important when you're trying to do something a bit different. They've been really enthusiastic, supportive, our investors, and a massive thanks to all the patients who are prepared to donate their samples and really help contribute to hopefully moving the needle in terms of generating cures. So I will end there pretty much on time. So thank you, Dennis.